Hello students, I am Ruchi Pandey, PGT Biology from JNV Agla Shopian, Jammu Kashmir. Today we will be dealing with the chapter excretory products and their elimination. This chapter will be covered in different parts. The first part will include the structure of kidney and nephron and how the urine is formed. Before going towards that, we need to understand why there is a need for elimination. As we all know that animals accumulate ammonia, urea, uric acid, carbon dioxide, water and ions like sodium ions which are produced by metabolic activities or by excess ingestion. These substances has to be removed totally or partially from the body. Now, for the elimination purpose, in the lower organisms, different structures have evolved from time to time, and the most advanced structure is evolved in the human, which consists of a proper, well defined system. It includes a pair of kidneys, one pair of ureters, a urinary bladder, and urethra. So we know that kidney is a reddish brown bean shaped structure located between the last thoracic and third lumbar vertebra, which is very close to the abdominal cavity. We have seen in the diagrams that usually left kidney is little higher than the right one because of more space being occupied by the liver on the right side. If we look at the kidney, then we will be observing that towards the center of the inner concave surface of the kidney is a notch-like structure. This notch-like structure is called the hilum, and this hilum is the point from where the ureter, blood vessels and nerves enter. Now, inner to this hilum is a broad funnel shaped structure which is called the renal pelvis. This renal pelvis will be having the certain projections, and these projections are called the calyx, or the future form is called the plural form is called the calyces. We can see that the outer layer of the kidney is usually a tough capsule in order to prevent the kidney from the shock or any kind of injury. If we look at the inner structure of the kidney, then it is divided into two parts. The one is cortex and the another is medulla region. The medulla is divided into a conical masses, and these conical masses are called medullary pyramids. This medulla, which is divided into different conical masses, they are projecting into the calyces. Now, the cortex is extending between the medullary pyramids, and the cortical region between the medullary pyramids is called the column of Bertini. We know that each kidney It consists of approximately 1 million nephrons. Nephrons, they are usually the structural and functional unit of kidney. As we all know that cell is the structural and functional unit of life. Likewise, nephron is the functional and structural unit of kidney. You can see that usually the nephron is divided into two parts. The one is the glomerulus and this another is this tubular part. Now, this tubular part is basically divided into three parts, which is called PCT, Henle loop, TCT, and the last one is collecting ducts. Let's discuss each structure one by one. First is glomerulus. Glomerulus is usually a tuft of capillaries which are formed by afferent arteriole. This afferent arteriole is a fine branch of renal artery. Next, there is a Bowman's capsule. It is a covering around the glomerulus. This Bowman's capsule, it is a covering around this glomerulus. Now, the Bowman's capsule is a doubled wall layer which consists of outer wall and inner wall. It is this inner wall which contains the filtration slits or pores through which the blood is filtered. The space between the two layers of Bowman's capsule is called the lumen. Now, this glomerulus and Bowman's capsule together they are called Malpighian bodies. Moving further, after the glomerulus Bowman's capsule comes the proximal convoluted tubule. So, we are dealing with this part. Now, let's have a look. This PCT, the diagram here is the one cell of the proximal convoluted tubule. It is lined by simple cuboidal brush border epithelium to increase the surface area for reabsorption. Now, the cells of this region, this proximal region, contains abundant mitochondria and food reserves in order to have active absorption and secretion. This proximal area, Henle loop and distal area is surrounded by a network of capillaries which is called peritubular blood capillaries. This peritubular blood capillaries, they are the part of efferent arteriole. We will discuss about this later on in this slide. The next part is loop of Henle. This is a hairpin loop-like structure that is in the medullary region. It consists of two parallel limbs. The first portion is called the descending limb and the another portion is called the ascending limb. It is joined by a U-shaped structure which is called loop of Henle. Moving further, this ascending part of the Henle loop is continued to the distal part and it is the highly coiled part of the nephron and it is very close to this renal corpuscle. Now, collecting ducts. It is basically the straight tube into which many DCTs open. Many ducts they converge and open into renal pelvis through medullary pyramids in the calyces. One very important thing is that glomerulus and tubular portion of the nephron they lies in the cortical region of the kidney, while the loop of Henle they lies in the medulla region. Now, the vasa recta, it is nothing but the peritubular capillaries which are formed by efferent arteriole. This is basically emerging from the glomerulus and called peritubular capillaries. A very minute portion of this runs parallel to the Henle loop and it is called vasa recta. Vasa recta is usually absent in cortical nephrons. So based on whether the Henle loop is short or long, there are two kinds of nephrons. The first kind of nephron is cortical nephron, and this includes the major portions of the nephron, means majority of the nephrons are of cortical nephrons. You can see here in the cortical nephron this loop is short, and in the juxtamedullary nephron this loop is long. So in the cortical nephron loop of Henle is short and they extends very little to medulla, while in another the Henle is very long and runs deep into the medulla. Now, after studying about the kidney and the nephron's structure, let's have a look how urine is formed inside the body. So, if we talk about urine formation, we will be talking about mainly three steps. The one is glomerular filtration, another is tubular reabsorption, and the last one is tubular secretion. So, as we can see that in this diagram, H2O is liberated out from the tubular part and reabsorption of water is taking place. So let's have a look how this is going to happen. So the first step is glomerular filtration. Why this is called glomerular filtration? Because it is carried out by glomerulus. So glomerular capillary blood pressure causes filtration of blood through three layers. The one is endothelium of glomerular blood vessels, this one. The another is glomerular basement membrane, and the next is epithelium of Bowman's capsule. Now we can see that here there is a basement membrane. So what happens is blood is filtered so finely through this filtration membrane that except the protein, all constituents of the plasma pass into the Bowman's capsule lumen. So we can see that this is glomerular space, this is endothelium, this is glomerulus basement membrane, and the blood is filtered through these filtration slits, and these are called the podocytes. So we know that On an average, 1100 to 1200 ml of blood is filtered by the kidneys per minute, which basically constitutes the one-fifth of the blood volume pumped out by each ventricle of the heart in a minute. It consists of three layers. The one is endothelium of glomerular blood vessels. The another is epithelium of Bowman's capsule, and there is a basement membrane. This epithelium layer, they are arranged in such a manner that they form the pores through which the blood can pass from one side to another, and it acts as a membrane for ultrafiltration. Now comes the tubular reabsorption. So initial filtrate which is formed is 180 liter, that means 125 ml per minute, but we know that urine released is 1.5 liter per day, so means almost 99% of the filtrate has to be reabsorbed by the renal tubules. So how this reabsorption is carried out? This reabsorption is carried out either through active or passive mechanism. Usually glucose, amino acids, sodium ions in the filtrate are reabsorbed actively, and nitrogenous waste are absorbed by passive transport. Reabsorption of water will also take place passively in the initial segments of the nephron. The last part is the tubular secretion. Tubular secretion is basically the phenomena of secretion of metabolic waste by tubular cells into the filtrate, like potassium ions, hydrogen ions, ammonia, and some waste like creatinine, certain toxins. So this basically form the tubular secretion, and a very important role in the balancing of acid base balance of body fluid. You can see here that from this diagram that in the proximal region of the nephron, selective reabsorption of glucose by active transport occurs, and then salts are reabsorbed by the diffusion. As a result, water is also reabsorbed due to osmosis, and the remaining fluid in the collecting duct will form urine, and that will be consisting of the concentrated urea, means less amount of water, and this finally leads to ureter. So this is all about how the urine is formed, how from the initial filtrate to that 1.5 liters per day of the urine is formed, and how the reabsorption and secretion plays a very important role in achieving this. In the latter part of the slides, we will be discussing with the functions of the different tubules and the control mechanism of kidney function. Till then, we have certain questions. After reading through this content, you can have a look on the questions. Thank you.